I am Rachna Deshmukh. I have recently joined as a biology teacher and today I will be teaching you absorption by roots. Now class, one very important thing. If I am not wrong, when you started studying science, the first chapter that was taught to you were parts of a plant. Now, if you remember, you had studied that a plant has a root and plant has a shoot. So, we are going to go into the details of the root, its structure, its function. So, it is nothing new. It's very simple and most of you must be knowing a lot of things that I am going to tell you today. Well, coming back to roots, what are the functions of root? First and foremost, you know plants are stationary. The roots fix the plant to the soil. Secondly, the roots absorb water and minerals from the soil within the soil particles, and then thirdly, they conduct it up to the stem. So if we talk about functions of the root, there are three main functions. First, anchorage, which means it fixes the plant to the soil. Second, it helps in absorption of water and mineral salts. So, absorption of water and minerals from the soil. And thirdly, it helps in the conduction of these absorbed substances to the upper parts of the plant, to the shoot, to the branches, to the leaves, to the flowers, everywhere. So this is clear. The chapter is titled Absorption by Roots. Now, why does the plant need water and minerals? This is clear. It is fixing the plant because it is stationary, it cannot move. But why does the plant need water and minerals? Simple. Again, you all know plants are the only living organisms on this earth that can fix the energy of the sun or fix the energy, radiant energy, into chemical energy, which is stored in the form of food. Or we say plants are the only living organisms. They are autotrophic. Auto means self. Trophic means nutrition. That is, they manufacture food. And what is this process called? This process is photosynthesis. Photo, in presence of light. Synthesis, manufacture of food. So, water is very important for photosynthesis. If you remember, water and carbon dioxide are the two raw materials that are needed for photosynthesis. So, water is one of them. Carbon dioxide we can get from the air. The plant takes carbon dioxide from the air. But water it has to take from the soil. So, this water has to be absorbed. Then secondly, minerals. Minerals make up the cell cytoplasm, various kinds of cell organelles, the various kinds of reactions that are taking place in the cell, form compounds, organic compounds for which minerals are needed. They are needed as catalysts, they are needed as cofactors of enzymes. That is, since plants are living, large number of reactions are going on in the plant body also. For that, lot of minerals are needed. Now, these minerals are absorbed by the plant either in the form of salts, let us say nitrates, carbonates, phosphates, sulfates, and simply as ions like potassium ion, like sodium ion, like chloride ions, etc. I am just giving you a few examples. So, these are the things that are needed by the plant and they have to be absorbed from the surrounding soil particles. So, this is one. Plant need water for photosynthesis. Secondly, you have learnt that water How does rain come? Water is given out by plants in the form of water vapor from the aerial parts of the plant, and the process is called transpiration. Transpiration, if we have to define transpiration, we say it is the loss of water in the form of water vapor from the aerial parts of the plant. And the purpose of transpiration is to cool the surrounding environment. And a very important function other than cooling is that once water is lost from the leaves, from the aerial parts of the plant, what happens? A deficit is formed, because of which more water is absorbed by the roots. So this is also called transpirational pull. It is a kind of suction force. So one function, water is needed for photosynthesis. Second function, water is needed for transpiration. Third function is, you all must have seen that during the morning time or evening time, the leaves of the plant, the flowers are erect. They are erect. This is because each cell of the plant body is turgid. Turgid means it is in a fully distended or extended state containing a lot of water. But sometimes if there is deficiency of water in the soil, or because of warmth from the sun during the daytime, during very high temperatures, more water is removed for cooling the plant body. As a result, what happens? The water content in the cell decreases. And water content in the cell decreases means the cell is no longer turgid. And opposite of turgid is flaccid. And we see the leaves that were standing erect during early morning times now droop down. Now droop down means the cells are flaccid, they are not turgid. As a result, they lose the mechanical strength. So third function of water is for mechanical stiffness of the plant. Right? And fourth, water is needed for various kinds of chemical reactions that are going in the body, and these things that are formed within the plant body have to be transported from one part to another. You have studied in your junior classes that leaves are the factories where food is synthesized. Now, if leaves manufacture food because they are green and they have chlorophyll, so photosynthesis is done in presence of light, in presence of green pigment chlorophyll. But what happens to the food after it is synthesized? It has to be translocated, it has to be transported to various parts of the plant. So the fourth function of water is the medium of translocation of these substances. So why do we, here I'll write why we need water. What is the need for water in a plant? Firstly, water is needed to perform photosynthesis. I have just now explained to you in detail what photosynthesis is. Second, water is needed for the process of transpiration to create a suction force, which we call as transpirational pull. Water is needed for mechanical stiffness of the plant so that the cells can be in a fully distended or extended conditions. We call it turgid. The plant cells are turgid. And fourth is it is needed for conduction or transportation of various substances, especially food that is prepared by the leaves, from the leaves to all other parts of the plant. Is that clear to all of you? Good. Now I will erase this and we will move to what are the processes. This is the main part of the chapter. That is, what are the processes that help in absorption. Basically, there are 5 main processes. First process is called imbibition. First process is called imbibition. Second process that occurs for absorption of water is diffusion. Third process is osmosis. Fourth process is active transport. And the fifth process, let us say is called turgidity, and opposite of turgidity is flaccidity. So now I will be going into the details of each of these processes one by one. Now first, imbibition. What is imbibition? Imbibition, now before I explain this, I am putting a question to you all. During the monsoon season, have you noticed that the drawers of cupboards, or let us say the doors of the house, swell up, because of which it is difficult to open them or difficult to close them? Sometimes the drawers get stuck. Suppose the dressing table drawer, it gets stuck. You can't pull it. Why does that happen? It is not a root. Simply it means that the living as well as the dead plant cells. Wood is nothing else but dead plant. So the living as well as the dead plant cells, they adsorb. See the spelling, it is different from absorb. Adsorb water in the upper layers. So we say this is adsorption of water by the surface molecules. Why? Because these are made up of hydrophilic. The word philic means like or affinity. Hydrophilic substances having affinity for water. So even if there is moisture around in the air, water vapor is there, they can absorb it at the surface. So that is imbibition. And very important process when we soak seeds. And suppose you soak seeds in a container and put the lid. You put water, put dry seeds and put the lid. After some time you will see that because of a pressure created due to imbibition, what we call as imbibitional pressure, the lid of the box suddenly opens. Because a high pressure is created because of this process, imbibition. So two examples I have given you. You have understood. This is something which we see in our day-to-day lives. It is not, science is not something very difficult to understand. The basic thing is you have to be observant enough to see things happening around you. And once you see things happening, you observe them, then you can go into the depth of it, why a certain thing is happening. So science can always be proved by experiments. So here imbibition you have understood. Imbibition is the process of adsorption of water by the surface molecules of dead or living plant cells. Then we move on to the next process, diffusion. Now diffusion, again I will give you an example in our day-to-day lives. You all must be having tea or milk or nimbu pani. What do? You add sugar, you add salt in it, and then you stir it. What happens? If you forget to stir it and you have your tea, when you take first 2 sips, immediately you can't feel the taste of sugar and you think either you have forgotten to put sugar or you have forgotten to stir it. Then you first thing, instead of going and putting more sugar, you stir your cup of tea, again taste it, and then if it's okay, otherwise you put sugar. What is happening when you are doing this? Suppose you add a spoonful of sugar in your tea or in your milk. Sugar settles down at the base of the cup. When you stir it, the molecules of sugar that were concentrated at the base now are spreading all over the tea that you have prepared or milk, and soon they are going to be equally or uniformly distributed in the whole cup of your milk or tea. So this is diffusion, which means movement of molecules of any substance, be it solid, be it a gas or a liquid, from a region where they were more concentrated, or we say from a region of higher concentration to a region of lower concentration. That is what is diffusion. Now here there is a point to be noted, that the two substances should be in direct contact of each other. There should be no separating membrane between the two. The two substances which are going to uniformly mix should be in direct contact with each other. So when we have to define diffusion, we say diffusion is the movement of molecules of any substance from a region of its higher concentration to a region of its lower concentration when the two substances are in direct contact with each other. So I have given you examples. Now, stirring of salt increases the diffusion. Even if you don't stir it and leave the tea like that or milk like that, or let us say you are preparing nimbu pani, you put the sugar and salt in it, and if you just leave it like that, over a period of time it will dissolve by its own, but the process will be slow. Stirring is making it faster. Same way, when you add sugar in water and you just raise the temperature of that water, it easily dissolves. So these two factors, that is stirring and rise in temperature, is helping the diffusion to occur at a faster rate. Now this you can't see with your eyes. So when you go to the lab, you will see this as a visual proof. I do not know whether you all have used potassium permanganate in your homes or not, but in the lab we do the experiment with potassium permanganate crystals. You take a beaker of water, put potassium permanganate in it. The potassium permanganate will, suppose this is a beaker of water. This is water. You put crystals of potassium permanganate. I would say potassium permanganate is just like the pink color that everybody uses during holy time. So you put that, what happens? It settles at the base. Slowly it starts moving. You can see the color moving. There is concentrated dark pink color at the base. Slowly it moves, and after some time we see that this dark color has disappeared from here, and we have a uniform solution which is pink throughout the beaker. In the fluid inside the beaker, the water inside the beaker is uniformly pink. So this is visual proof. That is, you can see how a substance from a region of its higher concentration to a region of its lower concentration takes place by diffusion. Another example I will give you. It happens in my class sometimes. Children carry deodorants and perfumes, and if you open a perfume bottle, first the teacher will not be able to smell it. But after a little while, the teacher can also smell it. How? Because diffusion has taken place. Molecules from where they were released have now mixed with the air of the room, and it has been uniformly distributed all over the room, so anyone in the room can smell the perfume. So that is molecules of a gas diffusing in air. Air is a mixture of gases, and perfume, when you release it is fluid when it is in a bottle because it has been put under high pressure, and when you push the nozzle, what happens? You are releasing the pressure, and when you are releasing the pressure, the perfume comes out and it mixes out in the air. So these are common examples of diffusion. Then we come to osmosis. Osmosis is also a kind of diffusion. The only difference here is that the two regions of different concentrations are separated by a semi-permeable membrane. The two regions of different concentration. Suppose I say this is water with salt dissolved in it, and this side we have pure water. Water, you know, is a universal solvent. Water is a universal solvent. Universal means everything, almost everything can dissolve in water. So this is water having salt molecules and on this side of the membrane we have pure water, which means there are lot of molecules of water this side. There are fewer molecules of water this side, and these dark blue circles are molecules of salt. Semi-permeable membrane is also called a differentially permeable membrane. When you were doing the structure of the cell membrane in a cell, you had studied that cell membrane is semi-permeable in nature, it is differentially permeable in nature, which means it has got pores and it allows only those substances to pass through it that are smaller than the pore size. And water can easily pass through the semi-permeable membrane because it is a solvent. So in case of osmosis, we say it is actually diffusion of water molecules from a region of its. The word its is very important. Wherever water is at a higher concentration, from a region of its higher concentration to a region of its lower concentration, when the two are separated by a semi-permeable membrane. So this is a region where we have more water molecules. So water will move from a region of its higher concentration to a region of its lower concentration. Is that clear? So it is nothing but, osmosis is nothing but it is a kind of diffusion, but the two substances I told you here have to be in direct contact and here they have to be separated by semi-permeable membrane. Is that clear? Now to explain this further, I'm just erasing all these processes that I have named because now I will be using the blackboard to make diagrams to explain osmosis to you. A very simple experiment is performed to prove that osmosis takes place using a thistle funnel. Now, you all know what a funnel is, but you may not know what a thistle funnel is. A thistle funnel is a very small funnel with a long, very thin and long stem. Very small funnel with a very thin and long stem, very narrow, like a straw, so that capillary action, water can rise in this tube of the funnel through capillary action, by adhesion, by cohesion, so that the water molecules stick to the walls of the tube. So this is thistle funnel. Now this thistle funnel is clamped on a stand. My diagrams are very rough just now, but basically you will find all these diagrams in the book. The only thing is you have to understand the concept that I am trying to explain. Normally I don't draw such bad diagrams. See, this is thistle funnel. This thistle funnel is immersed in a beaker of water. The mouth of the thistle funnel we tie a semi-permeable membrane. Now, semi-permeable membrane, a cellophane paper is also semi-permeable, and the cell membranes or plasma membranes of all kinds of living cells are semi-permeable in nature. We can use an animal bladder also. Anyway, we tie this. Then we fill this thistle funnel with say about 20% salt, or we can even use sugar solution. We fill the thistle funnel with 20% salt or sugar solution, and in the beaker we take pure water. Now two different concentration solutions are there. There are two regions of different concentrations. Now inside the funnel we have a solution. I am writing the word concentrated because I am comparing it with the solution that is in the beaker, and that is pure water. We have two different regions having different concentrations. Now inside the thistle funnel there is concentrated solution because it is 20% salt or sugar solution, and outside the thistle funnel we have pure water. So when we talk in terms of different regions, we can say this is a dilute solution or pure solvent. Better would be to say that this is pure solvent, and here this is a concentrated solution. Dilute and concentrated are comparative terms. Now, if you remember in the beginning I told you osmosis is movement of particles of water from where they are more. So, pure water has more water molecules, and here this is inside of the cell having 20% salt or sugar solution. This is outside of the cell having pure water. So, water will move from outside to inside. Now, inward movement. This inward movement or inward osmosis is called end osmosis. This inward movement is called end osmosis. Water always moves from a region of its. Lay stress on its. It is the key word. Or water moves from where it is at a higher concentration to a region where it is at a lower concentration, or a region of its higher concentration to a region of its lower concentration through a semi-permeable membrane. This is a must. Mentioning semi-permeable membrane in osmosis is a must. It is a key term. Semi-permeable membrane and its higher concentration of water where it is higher. Or if we are not talking in terms of water, then we talk in terms of solution. So it moves from a dilute solution to a concentrated solution. Always from a dilute solution to, and we always talk in terms of movement of water molecules. So this is osmosis. This is inward osmosis because we are comparing this to inside of the cell and this pure water to outside of the cell. Then in contrast to this, if there is outward movement of water, say the inside is more concentrated solution and the outside is, the inside is more dilute solution and the outside is concentrated solution. So the water will move from inside to outside because there is. Now I have explained end osmosis. Now coming to in contrast to this, what happens if there is a dilute solution inside the cell and a more concentrated solution outside the cell. The result will be movement of water from where it is more, that is inside the cell, to outside the cell. Movement of water from inside to outside the cell is called osmosis. Here it means there is more water inside the cell and there is less water outside the cell. So wherever there is more water, movement will take place from that region to where it is less. So here this is called exosmosis. Now to give you an example of end osmosis, you all, if you are observant enough, you all must have noticed that mummy soaks rajma or chana in water overnight or say for 6-7 hours. And what happens? After that we see that the seeds swell up. Why does that happen? Because the inside of the seed, those seeds are dry, there is less water, but the concentration of the substances, the solutes is more. Whatever organic food material that the seed contains, that is more and less water is there. So when it is soaked in water, water moves inside the cell, that is end osmosis. And that another reason for movement of water or swelling of the seeds is imbibition. That is, the surface of the seed absorbs water. So that was end osmosis. Then exosmosis I have explained. If water moves from inside to the outside, like I say, if we soak grapes in a concentrated salt solution, after some time we will see that the grapes shrink in size because there is more solute outside the grapes and there is less solute inside. So that means there is more water inside the grape and less water outside. So water from the grape moves outside and the grapes shrink in size. And in contrast, for end osmosis, if dry grapes, what we call as raisins, kishmish, if they are soaked in water, they swell up because water from outside enters into the raisin. The raisin is dry and has more concentrated solution inside. So these are day-to-day life examples of end osmosis and ex-osmosis. Now, another thing that I want to point here, that we were discussing this experiment on osmosis and I told you, explained to you regions of two different concentrations. Now, whenever any experiment is done, we always have to set up a control experiment for that. Now, what do we mean by control experiment? Control experiment means when we are going to take both these regions as same. Now, I would like to tell you one more thing, that whenever we do any experiment, we always have to set up a control experiment for that. Now, what is meant by control experiment? Control experiment means that it is used for comparison and the factor that is under study is missing in that experiment. Now, for example, when we are demonstrating osmosis, we have taken two different regions of concentrations. In the Thyssen funnel, there is 20% salt or sugar solution and outside it there is water. That is, there are two different regions of concentration. Now, in a control, inside the funnel also there will be water and outside the funnel also there will be water. That means the factor under study, the region of a different concentration is missing. So, we will see that there will be no rise of water in the Thistle funnel because same amount of water is entering the Thistle funnel, same amount of water is moving out of the Thistle funnel. So, there is no net movement of water in a control experiment. The level in the beaker won't fall, the level in the Thistle funnel will not rise. So, this is about control experiment. Now, this experiment on osmosis that I have explained, we can see different modifications in this. So, instead of drawing new diagrams, I will explain those different situations in this setup, this diagram that I have drawn over here. Now, in place of a cellophane paper, if we use a rubber membrane or a rubber bladder, rubber is impermeable. It is impermeable, means it does not allow the movement of anything through it. So, Thistle funnel has 20% salt solution. The outside beaker has pure water or pure solvent. Now, in this case, if there is a rubber membrane here, there will be no movement of water from outside to inside. So, the level here in the Thistle funnel will remain same. That was in the beginning. And the level of water here in the beaker will also remain the same. There will be no change in level because rubber is impermeable in nature. Now, instead of a rubber membrane, if we use a muslin cloth, what is muslin cloth? Malmal ka kapda, very thin material which is porous. And if again I would say, if you're observant enough and you have seen mummy working in the kitchen, you must have seen how she sets paneer. The water has to be drained and generally she puts it in thin cloth. So what happens? All this muslin cloth is fully permeable. Means all the water will come out. There will be nothing left in the Thistle funnel because there was solution here. The entire solution, because it is fully permeable, so solute as well as solvent molecules will come out in the beaker and we will see that the level of water in the beaker will rise and this will become empty. So these are three different situations that we have dealt with today with osmosis. First was semi-permeable membrane, then we use a rubber bladder, then we use a muslin cloth. And control experiment is very important. So I think I have discussed almost all the aspects of osmosis today. You read from your books, go through all the experiments and different options that are given, and you will get a clear picture of what has been explained today. Here I would like to point out, children, that you all should make notes. Either you pen down the important points or write detailed notes, because that is essential. And I will also be posting questions related on these 3 topics that I have taught you today: imbibition, osmosis, and diffusion. Solve those questions, make a separate notebook for that so that I can check those questions.